All right, hello, good students of AP Bio. I am coming to you from my cabin. I'm sitting outside right now, actually, on December 10th here. That's crazy. If you hear some weird sounds in the background, the ice is forming on the lake and really singing away, so it's kind of a cool sound. I don't know if the microphone will pick it up. Ultimately, today, we're going to watch this screencast. I don't have it linked yet, but this is going to be a screencast linked right here that I'm making currently. We will go through a worksheet, and the Jamboard that I will use you'll have access to. So I want to talk about what are the chances of making you or making anything on this planet through the process of meiosis, right? And so there were some key concepts from meiosis that we need to take away. First of all, it makes sex cells called gametes. That would be your sperm or your egg. It occurs in reproductive tissues or organs of organisms that use sexual reproduction. So anything that does sexual reproduction, it's going to have to go through meiosis at some point in its life. Most of your cells in your body, they do mitosis. They're just making copies of themselves. Only reproductive organs, that would be testes, ovaries, those types of things, those are the things that are going through meiosis, and they are making those things called gametes. Okay. Cells start with an even number of chromosomes. So your cells start out in meiosis with an even number of chromosomes. It always has to be even because you have to cut it in half, right? So even number of chromosomes, that full set is always called a diploid set. And at the end of the process, no matter what the organism is, the cells have half of the number of chromosomes that they started with. And that number is called a haploid number. And as you recall, hopefully from yesterday, the most important step is metaphase 1. That's how those chromosomes line up. And specifically in meiosis, they line up in homologous pairs. In mitosis, recall, they line up single file. So just a quick review from yesterday. I drew a little crappy diagram here. And you can see that with one homologous pair, that we have a couple of different things going on here. First of all, we have, I gotta get my pen out here, sorry. We have this concept that you can line up during metaphase 1. This is going to be metaphase 1 right here. During metaphase 1, you line up this way. So our centrioles are top and bottom here. So we're lining up kind of on that equator, right? And that would lead to, and we're skipping a whole bunch of steps here, but that would lead to two cells, each having a duplicated chromosome. Still looks like an X. And then after the second meiosis, after the second split, and if you need to go back and review meiosis, go do that, because I'm cutting a bunch of steps here and hoping that you realize, like, okay, so this is what the end product is going to look like. We're going to have two cells that look like this and two cells that look like this. So in other words, two are going to carry dad's information and two are going to carry mom's information. But we talked briefly about the fact that, well, what if they lined up opposite? What if the partnership, what if the pair lined up flipped compared to this beginning? What if they lined up this way? That give us different results. And the answer in this case is no. There's not any different results. We'd get either moms or dads, and that's the same that we got before. And that shouldn't be too bad of a concept. But what if we have two homologous pairs? I got a cleaner picture for that one. So if we have two homologous pairs, then what ends up happening here is you have the first possibility is they line up this way. Now realize they put the centrioles in this one on the side, so the poles are on the side, so that equator is in the middle here, remember, okay? So they paired up this way and they paired up this way. After metaphase, this would be metaphase 1, after the first split, and then into metaphase 2, they'd be lining up like this. Single file in metaphase 2, it's just like mitosis, right? And then in anaphase 2, the chromatids would get pulled apart. So like this guy would go this way, and this guy would go this way, and the whole thing would repeat basically throughout all of these cells here. But you're going to end up with either this or this situation here, with this right here, with that setup. Well, what happens if the bottom pair flips? What if they line up opposite? What if this bottom pair, and we look at that over here? So if that bottom pair flipped, do we get a different combination? And the answer in this case is yes, we do. It does matter. We end up with and I'll call them long and short, and dad and mom. We end up in this combination with a long dad and a short mom. Same thing here. And we end up with a long mom and a short dad here. Chromosomes are different than they were in these original sets here. So how many different combinations do we get? We get 4 combinations. Take a look at that. Pause the video if you need to and get a concept of that. Again, they are saying some phases are not shown here. They are skipping a whole bunch of steps here. But ultimately, realize that with 2 homologous pairs, you're going to get 4 possible outcomes. Recall that with one homologous pair, you're going to get two possible outcomes. No matter how you do it, you're going to get this one or this one. Even if you flip them, you're going to get that one or that one. With three homologous pairs, this gets uglier and uglier. If this is our original set, our original one here, okay, we'd line up all moms on the left and all dads on the right. And again, our centrioles are on the opposite sides here, but that's okay because we can visualize that, I hope. This is going to lead to, in the very end, those two cells being created, that type of variety. So there's two varieties that are made here. This one, what if we flip the bottom two? So what if we flip the bottom, the short pair here and the medium pair? What if we flip those two pairs? Well, what ends up happening is you end up looking like this then, right? This, if you look at this cell here, this one has the bottom two pairs flipped. They aligned differently. That's going to give you a different outcome then. In the very end of everything, that's going to give you a different outcome. Well, what happens if you flip just the middle pair? That's this one. What happens if you flip the top pair? That's that one. Or no, is that the middle pair? I don't even remember anymore. It's just the bottom pair. What if you flip just the bottom pair? So this is the bottom pair. This one is the middle pair we flipped. And this one is the bottom two pairs that we flipped. You get different outcomes each time. And I'm hoping that you can see ahead to that you can see that, okay, yeah, you split this and you split this and you split here, and then you separate the chromatids out in the second stages, and you're going to get these different outcomes. So we either have all long moms and all long dads, or we have combinations of different types here. Now, if you're really creative and you're thinking about this, you might wonder, well, all right, so we flip the bottom. What if we flip the top two? What if we flip the top two? And realize that if we flip the top two, I'm going to show you just real quickly here, that's going to give us the same exact thing here. So I'm going to give a long mom and a medium mom and a short mom over here. And then we have our short dad over here and our medium dad and our long dad over here. That is actually the same as another one on this list right now. And I hope you can see it. It's going to be the same as this one over here, right? It's going to be the same exact arrangement. We just have to mirror it. So it's like if the cell just kind of rotated around itself and turned around, and you rotated it, yes, you'd have blue long, blue medium, red short. Notice that over here we have blue long, blue medium, and red short. And that's the same exact thing. So that doesn't give you a new combination. That one doesn't give you anything new. So again, with three homologous pairs, how many different outcomes do we have? We have 1, 2, 3, 4, 5, 6, 7, 8. Okay. So that leads us to our table here. With one homologous pair, if you recall, we had just two possible outcomes. With two homologous pairs, go back to the slide if you need to, and in fact, I'll just do it real quickly here. With two homologous pairs, you ended up with one combination. Remember, these are repeats of each other. A second combination, a third combination, and a fourth combination. So 4 different types. With 3 homologous pairs, we ended up with actually 8. So we ended up with one combination like this, one like this, one like this, one like this. So we had a total of 8 combinations. With 4, what do you think that number is going to do? Well, it went from 2 to 4, 4 to 8. This one is going to go up to 16 different combinations. Your challenge for the worksheet that you got to do with this. So again, I'll open up the calendar here. This worksheet right here is right here. That's the table you need to fill in. So, and it says on the top, use the screencast-o-matic, follow the video until you see that table below. You got to figure out the math formula. So kind of like a y equals, x type of formula, except that we're going to have number of combinations. Your formula is going to be like number of combos equals, and then some type of math with homologous pairs. And I did give a hint on the worksheet that it is not, here's the hint right here in the middle, it is not 2 times the number of homologous pairs. It actually doesn't work out that way. So it's going to take a little bit more creative thought on your part. And then you're going to go through, and you're hopefully going to be able to answer these questions down below here. That's your homework for that. And then it talks about here, keep in mind on step number 3 here, keep in mind that this example ignores the process of crossing over. So I just want to briefly touch on crossing over. Crossing over makes this even more complicated because when those chromosomes align, when those homologous pairs align during like prophase and metaphase, when they line up, the legs of the chromatids cross over, and you end up having the areas that cross over can swap with each other. And that's a crazy concept because what they do then is that they mix and match like their own dad's and mom's DNA that they got in the first place, and then they're going to pass down something different. These on the outside here, when they get separated in anaphase 2, that's going to be just the same one as their dad got from them or whatever. But these have recombined, so they're called recombinant. They are recombined, they are recombinant. And it's going to be a term that we're going to have to start using in genetics. So these have recombined. They actually give us mixes and matches of dad's DNA and mom's DNA. So here's dad's DNA and mom's DNA that this person could pass down in their gametes. So crossing over even cloudies this even more because what have we not accounted for here? We haven't accounted for any crossing over. We've just said that mom stayed moms and dad stayed dads. You know, mom stayed moms and dad stayed dads, and that nothing changed down here. But in reality, all of these would have crossing over events. Crossing over is incredibly common. It happens somewhere around 99% of the time. It's not the rarity. It's just easier to learn meiosis without crossing over because it's kind of simple then, and we can do this math with it, and we can answer the questions on here, but then realize that crossing over makes this much more difficult. And then finally, you're going to have to look up Mendel's law of independent assortment and see how that connects to how these chromosomes independently assort, how they line up in the middle during metaphase 1. Remember, metaphase 1 is always the key. So, hope that makes sense. I hope that you have some success with that. If you don't, get a hold of me, and I will be glad to go over it with you. Take care.